Okay, so today we're going to talk about something that we've alluded to several times so far this semester. How do T cells recognize things, and how does those antigens get on the surface? And we're going to start with MHC1 antigen processing and presentation, but really the next two lectures are how do antigens or epitopes get onto MHC proteins from both intracellular and extracellular sources. And so we're going to be covering a little bit of both MHC1 and 2 just to highlight the differences, and then we'll go a little deeper into MHC1 specifics. Okay, so if you remember this experiment, Peter Doherty and Rolf Zinkernagel, who were at St. Jude's in Memphis and also in Australia, showed that T cells recognize both self and foreign antigens. And the way that they did this is they took a mouse that had the MHC-A haplotype, so all of the MHC in that mouse are MHC-A, and they infected that mouse with a virus. And then they isolated the T cells and said, could they kill, what kind of targets can they kill? And so they found that if they had MHC-A cells but no virus, there was no killing. And if they had the wrong MHC, MHC-B haplotype plus virus, the T cells also did not kill them. It was only when you had the correct MHC, in this case the MHC-A haplotype, and LCMV, that you would get killing and recognition killing of the target cells. This really started a debate. If you go back and read their paper, they argue that there is probably just one receptor, but they could not rule out the idea that there's two different receptors on a T cell, one that's recognizing the MHC and one that's recognizing the virus. But that becomes a little unlikely when you think about virus biology, where virus could just mutate, and so it would either downregulate MHC, so you wouldn't be able to see it, or it would mutate itself, so it would no longer present something on the cell surface. So this started quite a big debate, right? And they proposed this dual recognition hypothesis. And the hypothesis is that T cells make one receptor that recognizes self-proteins, and another that recognizes the foreign antigen, or in this case, the virus. This would be similar to a receptor that looked like an antibody that would recognize a shape on the surface of infected cells. And so if you only had recognition of MHC, in this case is shown here in purple, that there would not be T cell activation. On the other hand, if you had recognition of both MHC, here in purple, and the viral antigen, then those cells would be activated and kill the target cell. We now know this is wrong, and we'll go into why that is. But even up until the mid-80s, some scientists were still arguing that this could work, and with the accumulating evidence, it became harder and harder to explain how that might actually work. Now, part of that is that it required changes in the self-MHC protein that would have to happen to make this a feasible hypothesis. Because we know it's wrong, I'm not going to expect you to know the details of why it was wrong, but essentially it came down to Pam Bjorkman, who in 1984 was the first person, working with Don Wiley and others, to crystallize an MHC peptide complex. And then they used X-ray diffraction to get the structure of that. And they showed that the antigen is actually bound into the groove of the MHC protein between the two alpha helices on the surface. And so this really put to rest the idea that there's a two receptor hypothesis. Instead, there's one receptor that's recognizing both the MHC in terms of these alpha helices and the peptide that's bound inside the MHC. And it took until the late 90s for this to be shown conclusively how a T cell receptor does this. And two different people did this independently, Chris Garcia and David Garbozzi, and they both independently crystallized a TCR MHC and peptide complex using X-ray crystal structure to show that the TCR is recognizing both the MHC alpha helices and this foreign peptide that's bound to the MHC. And this finally put an end to this dual receptor hypothesis. Both Pam Bjorkman's work showed, no, it's probably not true, and then Chris and David really hit the final nail in the coffin saying, no, this is how a T cell receptor actually recognizes antigen. Now that put the dual receptor hypothesis to bed, but unfortunately, it sort of only put it to bed because that we know is how NK cells will recognize antigen. They're recognizing a self protein and then some aberrant self protein. So it still kind of exists for NK cells, but at least for T cells, that's not how they recognize it. There's just one receptor. So if you think about the goal of MHC presentation, it's really the ability to take peptides from any class of microbe and to present those peptides to T cells that then can be recognized by the T cell receptor. And because of that, then you have, you can activate appropriate T cell responses to control the infection. So it's recognizing things differently than an antibody. It's recognizing linear peptide structures. So the questions really are, how do you get the peptides? How are they generated? And that it's very different for class 1 versus class 2. We'll go over some of those differences today. And then how do those peptides get transported to the MHC and bind? And then how does the MHC get to the cell surface? And then we covered this topic a little bit already, but how does MHC polygyny and polymorphism ensure that you can have a response to virtually anything that's trying to infect you? Okay, so those are the questions I want you to keep in mind as we go through this lecture. So the first thing that we have to sort of separate here is, where do the antigens come from for MHC1 and MHC2? And we'll go through sort of the classical explanation of that and then the exceptions for each of those. So in general, peptides that are going to bind to MHC2 come from extracellular sources. So they come from cells taking up parts of their environment, putting them into endosomes, eventually degrading those peptides and loading them onto MHC2. For MHC1, it's a little different. You're typically trying to take antigens or peptides from intracellular sources, so from the cytoplasm, and putting those through the endoplasmic reticulum into MHC1. Okay, so in general, MHC2 is for extracellular antigens, and MHC1 is for intracellular antigens. Now we'll see that there are definitely exceptions for that. So when we talk about these two different types of MHC, they have a little bit difference. They look very similar in terms of structure, but there are a few differences, and that includes where they're expressed. So if MHC1 is important for CD8 T cell recognition and killing of an infected cell, you basically have to have MHC on every cell so that any infected cell can be recognized and be eliminated. For MHC1, there's a single MHC protein. Here you can see the alpha 1, alpha 2, and alpha 3 domains with the transmembrane tail. And that's complexed with a soluble protein that's very high abundance in the blood that is called beta 2 microglobulin. Now, in general, MHC1 is recognized by CD8 T cells. And so it's important for T cell killing. And that's the main job of CD8 T cells. They don't have many other jobs. And so therefore it's going to present peptides from intracellular sources. Or another way of saying that is it's going to present peptides from the cytoplasm, indicating that cell is infected and needs to be killed. Now, MHC2, on the other hand, has a much more limited expression. it's only expressed on professional antigen presenting cells. Initially dendritic cells, but then later macrophages, B cells, and T cells can all act as antigen presenting cells. Now in contrast to MHC1, which has a single MHC protein with beta-2 microglobulin, MHC2 has two MHC proteins, an alpha and beta chain. And this has some implications for the number of different MHC that can be expressed on the surface, including mixing and matching of different alleles. The MHC2 is typically recognized by CD4 T cells, and so it's really the antigen presenting cells are sampling the environment, and then they're going to present it to CD4 cells so that they can become activated and direct the response. Okay, so it's presenting extracellular derived peptides. And that really boils down to the different jobs that these T cells have. Okay, CD8 T cells are that become activated, become cytotoxic T cells. They have one job: go out and kill infected cells. So mostly they're going to be looking for cells that are presenting antigens from intracellular sources, indicating that cell is in fact infected. CD4 T cells, on the other hand, are not necessarily going to be killing target cells. Their job is more directing the response. And so they're going to be activated, by antigens presented from extracellular sources. And those cells will then activate the macrophages, provide help to B cells, provide help to the CD8 T cells. So they're not looking to go out and find infected cells and kill them, so they can recognize the extracellular peptides. Now, the question really then was, how are these peptides generated, or what do you have for recognition? And this is about the same time as Pam Bjorkman's work on the crystal structure of the MHC, but Pippa Merrick and John Kapler at the University of Colorado decided to use a little bit of biochemistry. And they had cells that expressed empty MHC proteins. And so we'll go into it, but essentially they lacked the transporter that would normally transport peptides into the endoplasmic reticulum for binding to MHC. And therefore, they could just load these MHC on the surface, okay? And the experiment they did is they immunized mice with a protein, in this case ovalbumin, as the antigen, and they isolated the T cells. They asked what kind of cells could those T cells recognize. And so if they just had the cells only, there was no recognition, okay? If they had the cells and they just added the whole protein, also was no recognition. But if they pre-digested the protein with proteases and it sort of broke it up, then those peptides could be presented on MHC proteins, and that allowed those cells to be recognized. So it was really showing that if you digest up a protein, then you can have the epitopes. And so the conclusion from that is it's linear epitopes that are presented on the MHC. And so when you look at the difference between T cell recognition and antibody recognition, antibodies bind to structures or shapes, okay? They're going to bind to parts of a protein that may or may not be contiguous in terms of the amino acid sequence, but they're brought together during protein folding. And so those, by definition, have to be on the surface of the protein. But oftentimes, the epitopes that are presented to T cells are from linear sequences of amino acids that are buried inside of the protein, okay? And so for those to be liberated, you have to have a digestion or breaking down of that protein. And then the epitope can bind to a self-MHC protein that then goes to the surface, and that's what the T cells can recognize. Okay, so there's a fundamental difference in the way that T cell receptors and B cell receptors recognize antigen. BCR bind conformational epitopes, TCR bind linear epitopes. So the question is, if this is necessary for generating peptide sequences that can bind to MHC, how do you make those? And so the first question is, how do we get those peptides? How are they made? Well, in order to understand that, we sort of have to understand how the peptides bind. There's a slight difference between class 1 and class 2, and we'll talk more about class 2 in the next lecture. But essentially, for MHC1, it is a very limited length of a peptide, typically between 8 and 10 amino acids. There's a few examples that are a little longer or a little shorter, but in general, they're short peptides. And that's because the ends of the peptide are closed, or the ends of that MHC binding groove are closed, meaning the peptide can't stretch beyond that. That's different than MHC class 2, where the peptide can be quite long. It's typically 13 up to, I've seen them up in the 30 amino acid range, because the ends are open. The ends of the MHC binding groove are open, and so that the peptide can kind of slip back and forth until it finds its best binding affinity, and that's what is then presented. Now that has to do with the way that these peptides are generated. MHC class 1 is a much more regulated process, where MHC class 2 is sort of a partial digestion as things are taken in from extracellular sources. Okay, now one thing that I should mention is that the peptide is an integral part of the MHC structure. And if you don't have a peptide bound to that MHC, it's quickly internalized and degraded. So it's important for, if there's MHC on the cell surface, that there is a peptide bound to it. And so we'll talk about how you're always keeping a peptide there and ensuring that there's something to be presented to a T cell. So if we sort of break down these two pathways, we have the MHC class 1 and MHC class 2 pathways. And so if we start with the MHC class 2, you have a antigen presenting cell taking up some extracellular antigen. This could be a whole bacteria or a whole virus. It could be parts of the bacteria or virus, or fungi for that matter. It's just sampling the environment. These peptides are brought in the endocytic pathway, and they fuse with what's called a phagolysosome. This is just in phagocytic cells, you have lysosomes that contain things that will start to break down those proteins into peptide fragments. At the same time, you're synthesizing MHC class 2, and this is going into the exocytic compartment, and it's actually a specialized exocytosis pathway called the MHC2 compartment, and this will fuse with these phagolysosomes, allowing these peptides to bind to MHC class 2. When that happens, that then goes to the cell surface and can be recognized by T cells. Okay, so MHC2 sort of have an endocytic and an exocytic pathway merging, and as those peptides are being brought in, they're degraded and then loaded as the MHC2 is on the way out of the cell or to the surface of the cell. Now remember, for MHC1, you're really surveying for infected cells, and so you're looking for stuff that's already in the cytoplasm. In this case, we have some viral antigens, and this is a much more regulated process, and that these antigens are fed into a protein complex called the proteasome. The proteasome is a multi-subunit protein complex that is taking peptides derived from these pathogens in, cutting them typically 8 to 10 amino acids long, and then feeding those into the endoplasmic reticulum. It's here, much earlier in the pathway, that MHC1 epitopes are bound by the MHC1 protein, and as they bind, they cause a conformational change, which allows this to be released to the cell surface. Okay, so the source is different, but also how those peptides are chopped up is different. MHC1, it's very regulated, short 8 to 10 amino acids, by the proteasome. For MHC2, it's sort of just digestion with proteases, and so you get much longer fragments, and so that has to bind to MHC2 differently. Okay, so the first thing that we have to think about then is how do these peptides that are generated from the proteasome get into the endoplasmic reticulum? And there is a dimeric protein called TAP. And this is the transporter associated with antigen processing, okay? And its job is to take peptides that are generated from the proteasome and transport those into the endoplasmic reticulum where they can bind to newly made MHC1. Okay, so the first thing is that you're going to degrade the proteins from viruses and intracellular bacteria in an infected cell, and then transport those into the ER by the TAP protein. And then once those are bound, the MHC1 peptide is transported to the cell surface, and that's where it can then be recognized by CD8 T cells. So let's talk about the degradation. So the proteasome is this fairly large complex of proteins that exists in the cytoplasm. It is actually loosely tethered to the transporter, so that all the peptides you generate are going to be fed directly into the endoplasmic reticulum. And there's really two core parts of this, or two subunits. The first is the 20S core protease. That is the catalytic portion, contains the protease subunits that are chopping up peptides. The 19S cap, or sometimes called the regulator, is responsible for binding to proteins that need to be degraded, so it recognizes them. So in the core, you have three types of enzymatic activities, and these are mediated by many different subunits. You don't have to know which subunit does which, but you can see that they typically cleave after either triptych or chymotryptic, meaning a positive charge or these sort of bulky amino acids, or they can cut after a negative charge, particularly glutamic acid residues. Okay, so do I care that you know all the things that it's going to cleave after? No, there's just a number of different amino acids that it has a preference for, and this results in cutting it into short amino acid fragments. Now, in terms of the regulator cap, it's going to bind to proteins that have been ubiquitinated. And this is if you remember from molecular biology or cell biology, that this is a way cells use to label old proteins or proteins that it's trying to downregulate or misfolded proteins. And so these proteins are basically recognized by the proteasome, and they're broken up and leading to recycling.
I had a question on the previous slide.
Yeah.
One more actually. When you showed the chaperone proteins, is the chaperone proteins assisting the peptides?
We'll talk about them in just a second.
Okay.
Because they're really important for folding MHC and then holding it open.
Okay, thanks.
So we will go into that in just a second. Okay, so this is just a brief review of ubiquitination. So most systems you have three ubiquitinases. There's an E1, E2, and E3. And the way you can think of this is E1 is the first step. You have some protein, E1 ubiquitinase, and it gets activated by ATP, and in doing so will transfer a ubiquitin to itself. So this becomes activated. Now the second enzyme in that pathway is the E2 ubiquitinase, and its job is to actually transfer the ubiquitin from E1 to the target protein. Okay, so the E1 is been activated with ubiquitin. E2 will come and steal that and put it on a target protein. The E3 ubiquitinase is responsible for which proteins are going to be recognized by E2. So E3 is really just bringing in target proteins to be recognized by E2 and transfer that ubiquitin. Once there's enough ubiquitin on the surface, then the proteasomal cap can recognize the ubiquitin and feed that protein into the proteasome. Okay, so ubiquitination is a regulated process. There are many different ubiquitinase pathways. E3 provides some specificity. The E3 will only recognize different types of proteins, but you have several different E3 ubiquitinases that can essentially regulate lots of different intracellular proteins. Okay, so we've gone through the steps, but essentially, if you think about this, what this means is that under normal conditions, the proteasomal degradation of proteins requires ATP because you require it in this first step to activate the E1 ubiquitinase. So if you don't have ATP, then this process shuts down. Okay, so under non-inflammatory conditions, your cells are doing this all the time. And so the proteasome is degrading self-peptides. And a lot of that is just for recycling. So ultimately, these are broken down to individual amino acids and reused to make new proteins. But that changes during inflammation. So inflammatory cytokines like IL-12 and type 1 interferons will cause the cell that is infected or has things in its cytoplasm to change the core proteins and the cap proteins of the proteasome. And so during inflammation, you get a change to become the immunoproteasome. Now in this class, I'm not going to ask you which subunits change. You should just know that in fact, they do change. And both the cap and the core proteins change. Now, the difference here is that in the immunoproteasome, under inflammatory conditions, the cap no longer requires recognition of ubiquitin. It's just requiring recognition of any protein. And so if you think, well, why would that be? Well, in an infected cell, in which you're replicating lots of, let's say, viral nucleic acids in the cytoplasm, you don't have a lot of ATP. And so if you just depleted ATP and you required that for labeling of proteins for feeding into the proteasome, you could just efficiently block degradation. So during inflammation, you no longer require ubiquitin, so you don't require ATP. The second part of this is the changes in the core proteins. And these actually don't change, excuse me, how it cuts. It actually just changes the efficiency. And so it makes it more efficient. It also binds it strongly to the TAP protein so that anything that's generated from the immunoproteasome is fed directly into the cytoplasm. So I sort of went over this. Why is it important to not need ATP for immunoproteasome degradation? And the answer is during infection, you often lose intracellular sources of ATP, and so you couldn't really do that efficiently. Okay, so now you've degraded the peptides in the cytoplasm. And how do you get those into the endoplasmic reticulum so they can bind to MHC1? And so the first thing is, well, there is a transporter. It's called TAP, transporter associated with antigen presentation. And so any peptides that are generated are bound by the TAP proteins, and they're taken from the cytoplasm into the lumen of the ER. There's a TAP1 and a TAP2. I just want you to know that it's dimer. And it has, when the crystal structure of this was found as it has sort of a hole in it. So it really isn't a transporter. It's feeding these peptides of 8 to 10 amino acids long into the ER. So the question then is, if I didn't really cover it, the TAP2 protein actually has an ATP binding cassette. And it's sort of like a G protein coupled receptor in that you have exchange of ADP for ATP that activates it. So how might you overcome the ATP requirement for TAP?
Open the channel.
Okay, it will open the channel, but by the immunoproteasome binding to it, it holds that channel open. And so you don't get ATP being converted to ADP and sort of closing of the channel. Okay, so the immunoproteasome doesn't, during inflammation, the immunoproteasome doesn't require ATP, and TAP also doesn't require ATP. But an easy question on exam would be, during non-inflammatory conditions, how would you regulate proteins being translocated? And that's because you have ATP, so you can still transport them even when there's no inflammation. Okay, so there was a question about these chaperone proteins. And the issue here is that you have to hold MHC1 open, because once the peptide is bound, the ends are closed and it's locked inside there. And so it has to be first synthesized. And so when it's synthesized, it's actually, the folding is directed by this general ER protein called calnexin. Calnexin is just making sure that the MHC1 is folded correctly and it's holding it there. Okay, that's a general chaperone protein that also helps fold many other proteins. And the second step is that then you have proteins that come in, and their job is to hold the peptide binding group open and put that MHC next to the TAP proteins. Okay, so the first protein is tapazen, and that binds to TAP and MHC1 and puts them next to each other so that the peptides coming through TAP can bind to MHC1. The two other proteins, calreticulin and ERP57, calreticulin is more specific for MHC, but ERP57 is just a general endoplasmic reticulum chaperone protein. Their job is to hold the MHC open so that peptides that come can actually bind in that peptide binding groove. Now, under non-inflammatory conditions, most of what's coming through the TAP proteins are what are called DRIPs, or defective ribosomal products. They're sort of, another way to say it is misfolded proteins. And John Udell showed that this is a major source of the peptides that are actually put through the TAP transporter. But you also have normal proteins, and generally this is older proteins or proteins that are being downregulated that have been labeled with ubiquitin. Remember, under inflammatory conditions, you don't have to have ubiquitination, and so it's really anything that's in the cytoplasm can be transported. So now you feed these peptides through the proteasome, and those are degraded and transported by TAP into the ER, and they can bind to MHC1. When it does that, it causes a conformational change in MHC1, and the chaperone proteins don't bind it anymore. It releases ERP57 and calreticulin, and it no longer binds to tapazen. And so the default pathway is then it just is transported to the cell surface. Okay, so there's 4 steps here. You synthesize it, you hold it open and put it next to the TAP, you transport the peptides that are degraded by the proteasome, and then it's released to the cell surface. Now, because of this turns out to be sort of a point in antigen processing and presentation that many viruses and some bacteria can block. And we'll come back to this when we talk about specific diseases and how they're the viruses are still recognized. But if you look at proteins that are expressed by viruses to block this, there's a number of steps that they can do it. The easiest one, and particularly herpes viruses do this a lot, is to block the transporter. Okay, so cytomegalovirus, herpes simplex virus express these proteins that essentially just prevent any peptides coming through TAP. The other way that you can do this is to say, well, I'm going to block the MHC associating with TAP. Okay, and so if it does that, then you get far fewer peptides loaded because they're just not right next to the transporter. Okay, so adenovirus will do this with the E19 protein. It blocks tapazen binding to MHC1, and so you can't put it next to the TAP. Another way you can do this is to dislocate the MHC from the transporter and actually feed it back in through another sort of degradation pathway, the Durlin pathway. And so the MHC1 is being degraded. It's essentially, the US11 is forcing this through Durlin. Once it goes into the cytoplasm, it's just like any other protein. It is then fed into the proteasome or immunoproteasome. Okay, so viruses are very good at this, and then typically it's very large viruses, pox viruses, herpes viruses, adenoviruses, viruses that have a large genome that have the space to contain immune modulating proteins.
So these proteins have evolved the function of not only doing this particular blocking something, but also getting into the ER?
Well, so remember, the viruses are being made in the ER. And so for example, the ICP47 from cytomegalovirus from herpes virus is actually being in the cytoplasm and is blocking TAP on the cytosolic side. The CMV protein US6 is actually a membrane-bound protein, so as it's being synthesized, it's being fed into the endoplasmic reticulum, where it's going to bind on the ER side of TAP. So was that your question, or were you guys saying about feeding MHC1 into the cytoplasm?
No, that answered it. Thank you.
Okay. So the question then is, how would this affect recognition by NK cells? So remember, NK cells have sort of a two receptor recognition. They have a kill and a don't kill signal. The kill signal is typically a protein that's upregulated during inflammation, and it can be almost, there's a wide variety of ligands for it. The don't kill signal is from a inhibitory receptor that typically recognizes class 1. So in the first case, if there's no peptides, remember, in order for this to be released, you have to have peptide binding. Without the peptides, it's unstable, it falls apart and gets degraded. If you're degrading MHC, in this case, then there's no MHC on the surface. And so an NK cell will come along and see a cell that doesn't have MHC on the surface, and so you don't get the don't kill signal. So the cells that do this are still able to be recognized by NK cells. The lack of MHC that would normally provide a don't kill signal is gone, and so therefore the NK cells says, aha, this cell's infected, I need to kill it.
Dr. Blattman?
Yeah.
So you mentioned that during inflammation, the proteins don't need to be ubiquitinated. And so you kind of said that anything from the cytoplasm is going to get removed. How do the cells prevent themselves from tossing out something really important, or is it just kind of risk it?
Well, if you think about, let's say you're a cell, and you're infected. You've sensed that by your innate receptors. You've gotten inflammatory signals from cytokines. And so you're pretty sure, okay, I'm infected. Your job is to actually get killed at that point. And so you don't really care if you're feeding out some of your own peptides or proteins on the cell surface. You're basically trying to put anything you can for T cells to see it. Now, in a virally infected cell, the virus is basically hijacking almost all of the cell's replicative ability and protein synthesis for making more virus. So the vast majority of what's there is virus anyways.
Okay, thank you.
Okay, now we have to talk about something that was a big thorn, because we knew that you make T cell responses to things that are not inside the cell. For example, viral glycoproteins, those are on the surface of cells. So how are you making responses to that? And Mike Bevan at the University of Washington was really the one who first showed this. And remember, this is before we knew how T cells were actually were recognizing antigen. So it was not clear how this worked, but we now know that, in fact, this does happen, and it's very important. And this is a little bit hard to understand. So you have two variables here. One is the MHC-A, or the MHC background, or the MHC haplotype. And the other is the strain of mouse. And so in one case, you have black 6 mice. In the other case, you have BALB/c mice. And they have different proteins, okay? And in addition to difference in MHC, they have difference in their background proteins, okay? And so in this case, what Mike did is he immunized mice, black 6 mice, that had MHC-A and B. Okay, so they were MHC-A haplotype and MHC-B haplotype. And he immunized them with an antigen. In this case, the antigen are cells from BALB/c mice, or strain 1, that only had MHC-A. Okay, and so what he was looking for is, can I get recognition of strain A peptides or strain A proteins on strain 2 MHC-B? Okay, so if you have strain 2 with MHC-A and B, those are not recognized because there's no antigens here. This is just self. So they don't recognize self with self proteins, self peptides on self MHC. However, they do recognize strain 1 proteins, or strain 1 peptides on the strain 1 MHC, okay? So they have the MHC-A, but they also have the foreign peptides on there. That's an expected result. What was not expected is that you could recognize strain 1 peptides on the strain 2 MHC. So the only cells here that have MHC-B on them are in the recipient, but you're recognizing peptides that are only derived from the donor, or the antigen. And this, then he said, how do you get a response to these foreign peptides on self-MHC? There has to be a mechanism for taking up antigens, feeding them into the cytoplasm, and then loading those onto self-MHC. And it turns out that that's actually a very normal thing, where you're taking exogenous antigen and you're feeding it into, it's taken up in the endosomal pathway, and then it's being fed by sort of a reverse tap. In this case, it's called the Sec61 protein. And it's taking these proteins from the endosynthetic pathway and putting them into the cytoplasm, where then everything else happens. They're fed into the proteasome. Those peptides are put through TAP, loaded onto MHC1, and then make it to the cell surface. Okay, so there's really only one extra step, and that's feeding these exogenous antigens into the cytoplasm. And that's done by the Sec61 protein. Okay, so its cross-presentation is just one extra step to taking an extracellular antigen and making it a cytoplasmic antigen that then you degrade and put it into the class 1 pathway. Now, the reason that's important is because most of the time, if you're talking about activation of a CD8 T cell, it's not necessarily going to be on an infected cell. It's going to be on a dendritic cell that has sampled its environment, gone to a lymph node, and presenting antigens from its environment. And so cross-presentation is really for activation of naive CD8 T cells. It doesn't really work very well for a cell to come along and kill it. So the presenting cell, usually, particularly in the beginning of a response, is a dendritic cell that has taken up this extracellular antigen, fed it into the cytosol by Sec61, or often called retrotranslocation, and then it's degraded by the proteasome. It is then fed through TAP and loads on MHC class 1. Now, once the antigen presenting cells do this, they're not killed. Their job is to activate CD8 T cells. So naive CD8 T cells are not going to kill. They're just being activated. Once they become activated, their job is to leave the lymph node, go out in the periphery, and look for any cytosolic pathogen or any infected cell. Now, this is where you have the classical pathway, where you have an infected cell, and it's taking intracellular pathogen proteins, degrading them on proteasome or immunoproteasome, feeding those through TAP, loading on class 1, and presentation at the cell surface. These effector CD8 T cells then recognize this on the surface of any cell and will kill it. And we'll talk about how CD8 T cells kill infected cells in the next section of the course. But there's a fundamental difference for priming or activation of a CD8 T cell response. It's mostly by cross-presentation, whereas the effector arm of the T cell response, or once it becomes activated, then it's recognizing through this classical pathway of just cytosolic presentations. Okay, so that's sort of MHC1 processing and presentation in a nutshell, but then we come back to this idea of polygyny and polymorphisms, and how is that affecting which peptides are being presented? Because different MHC will present different types of peptides. So the first thing we need to know is, how is the genome or genomic organization of MHC organized? And so it's actually very similar in mice and humans, where they typically tend to co-segregate. Not always, but this is a fairly tightly organized MHC loci, and it contains MHC1 and MHC2 genes. So here in humans, you can see the MHC2 genes. The ones that are presenting it are DP, DQ, and DR. And then there's some ones that help with loading that we'll talk about in the next lecture. And it's very similar in mice. You have sort of the H2 alleles. So it's just the histocompatibility alleles for mice. In humans, we call them human leukocyte antigens. It's organized the same way. You have the MHC1 and 2, right? So you have H2K, H2D, and H2L. And then you have IA and IE. And these are MHC2. In red are the MHC1 alleles. Associated with that are the TAP proteins and the proteasomal subunits that are upregulated during inflammation to form the immunoproteasome. So these are all one loci, either in humans or mice or other species. And so when we come back to the idea of polygenic genes, these are all codominantly expressed. So if you have one chromosome that has all of the MHC or MHCA haplotype, and you have another chromosome and it's all MHCB haplotype, then you're going to express A and B. If you have the MHCK haplotype or the MHCM haplotype, you'll have both K and M alleles expressed. Okay, so they're co-dominant. Now, when individuals that have different MHC alleles are mated, then you can have, essentially those are going to, most of the time, just segregate together by chromosome. So this individual got both the green and blue alleles, blue from the father and green from the mother. Their sibling got the red and yellow, yellow from the mother and red from the father. And then the other siblings have the other combinations. So siblings may or may not have the same MHC alleles. And in general, I have 4 brothers and 2 sisters, and none of us have the same MHC haplotypes. Okay, but it's all co-segregated from the mother and father. Now, why do we do this? Well, we talked about several of the reasons for polygyny, meaning you have several of these genes expressed, right? So for MHC1, go back to our loci here. For MHC1, you have HLA, A, B, and C. So you have 3 MHC1, but then you have 2 chromosomes, so you actually have 6 potential different MHC1. Mice, they have H2K, H2D, and H2L. And so mice also have 6 potential MHC on their surface. Okay, so that's polygyny, where you have many different versions of the genes that are potentially expressed. Polymorphisms is that at each one of those loci, you can have a different version. So everybody has, or most people will have HLA-A, but you can have AO2, AO3, AO4, AO5. Those are just different polymorphisms of the H2A allele, or sorry, the HLA-A allele. And so in any different individual, you have lots of different copies of the gene expressed. And in the population, there's lots of different versions of each of those copies, okay? And that ensures that at an individual level, you're very likely to be able to present something to a T cell. And at a population level, that some individuals are for sure going to be able to present something to a T cell. Okay, so those are the 2 main properties of MHC. And the question is, how does that actually work? Well, when you look at MHC variability, you look at where are the differences in the MHC alleles, the vast majority of those differences occur in the alpha 1 and alpha 2 domains. Okay, this is just a sequence, residue sequence. But when you look at where those mutations are, they're actually in the peptide binding part of the MHC. And so this affects which peptides can bind to different MHC alleles. So they're not all the same. When you look at the variation, this is actually from an older version. We now know that there's many more than this. There's something like, up in the tens of thousands of different MHC alleles in the human population. You can see that they all have a tremendous different number of alleles that could do this. And so a given individual has 2 MHC or HLA-A alleles, okay? But in a population, there are hundreds and up to thousands of different HLA variants. So that allows you to recognize in an individual, you have a high chance. In a population, it's a virtually sure chance that something is going to be recognized. So these differences between alleles are really primarily in the peptide binding part of it, or the peptide binding part of the MHC-A. Sorry, they're in the peptide binding part of MHC-1. And so when you look at the different peptides that are binding to different alleles of MHC-1, in this case, let's say that this is a human, let's say it's HLA-A. And it's allele 1, so it's typically a star HLA-A-O-1. Okay? When you look at all of the peptides that bind to that, they have a strong preference for a bulky side chain at the 5th position. So here's 1, 2, 3, for 5, and a strong preference for a leucine residue at the 8th position. Okay, so these are different peptides from influenza, CMV, Epstein-Barr virus that are presented on HLA-A01, and they have sort of similarities in their anchor residues. These are the parts of the peptide that are binding to the MHC. The rest of it you can see is very different. So it's really the anchor residues or what's binding. If you have HLA-A02, here we just call it allele 2, it has different anchor residues. There's a strong preference for bulky tyrosine residue at position 2, and a strong preference for a hydrophobic, sort of smaller amino acid at position 9. Okay, so those are the anchor residues for allele 2. And so peptides from hepatitis C virus, from the deadly ovalbumin protein, measles or coronavirus, they all have this sort of motif, and so you can present peptides on allele MHC HLA-A02 that have these anchor residues. Okay, so the differences between the alleles just mean that they have different preferences for anchor peptides. They don't change, or they don't have a preference for what's pointing out of the MHC towards the T cell receptor. They only have different preferences for what's going to bind to the MHC. So if you go back into biochemistry, right, when we look at peptide bonds, where there's a carboxy terminus and a amino terminus, those form a bond, then every other side chain is pointing in the opposite direction. Okay, and so if they're pointing into the MHC, it's an anchor residue. If it's pointing up out of the MHC, it's a TCR contact residue. Okay, and so the MHC will use this geometry to bind one side of this peptide sequence through what we call anchor side chains, and then force those other side chains to stick out of the MHC to be recognized by the T cell receptor. And so in any peptide on an MHC, you have anchor residues and you have TCR contact residues. And so when you look at, here is part of, it's actually a paper from David Garbozzi. Here is the part of the TCR. So it's the CDR3 from the TCR beta, excuse me, the CDR3 from TCR alpha. In this case, the CDR1 from the TCR alpha also contributes a little bit to binding. But remember, the CDR3 is the part that sits right over the peptide, makes the most contact with the peptide, whereas the CDR1 and 2 are typically binding to the alpha helices of the MHC. So when you look at what's being recognized here, you have the side chains, tryptophan or glutamic acid, that are pointing down into the MHC. Those are the MHC binding side chains. And the part that's facing out, here an arginine, here's a serine, that are being recognized by the T cell receptor, and typically by the CDR3. Okay, so this is, again, going back to the crystal structure that David Garbozzi and others figured out, is that here's our MHC protein. So here's one alpha helices, here's the other one, and then you have in yellow the peptide that's being bound to that. That's being presented to the T cell. It's then being contacted, this surface is being contacted by the CDR regions of the T cell receptor. Okay, so in cartoon form here, you have a T cell receptor, the TCR alpha and beta are contacting both the MHC and the peptide that's being presented. When you look at what is actually being recognized here, you can see that the CDR3 in yellow are overlaying right on top of the peptide. So This is sort of a silhouette of, if you were to look straight down, if you were looking straight down this way at what is being recognized, you're looking at where do these different CDR3 overlap with the surface of the MHC. You can see that this yellow peptide here is right underneath the CDR3 alpha and CDR3 beta parts of the T cell receptor. Whereas the CDR1 and 2, for both alpha and beta, tends to be more binding to the alpha helices of the MHC itself. So there are sort of different parts of the T cell receptor have different jobs. CDR1 and 2 are recognizing the self MHC, and CDR3 is recognizing primarily the foreign peptide. Now it turns out that the more immunogenic peptides are typically have hydrophobic TCR contact residues, where what you're actually doing is you're forcing something that normally wouldn't want to be exposed to water to be pointing straight out of this protein. And so it's sort of backwards biochemistry that you're trying to force presentation of hydrophobic TCR contact residues, and that is actually contributes a lot to T cells recognizing this. Okay, so it looks like we went a little fast, and so I want to go over sort of the questions, things I will ask you about on an exam. So the first thing is, go back to our questions that we started with in this lecture. How do you generate the peptides that are bound by MHC proteins? So that's both by the proteasome and then also the immunoproteasome during inflammation, and they're transported by the TAP proteins. And then the second question is, once you do that, how do those peptides in the ER bind to MHC and allowing that MHC to get to the cell surface? So you should know what the job of tapazin is. That's going to bind to TAP and MHC. And then calreticulin and ERP57 are going to hold that MHC open until a peptide binds. You should also know if it's exogenous antigen, how is that brought into the cytoplasm and then fed, right? It's fed through this pathway back to the proteasome and re-transported by TAP. So this is the Sec61 pathway. And then I'll probably ask you something about how does polymorphism contribute to binding to different MHC peptides, right? So you're really changing, polymorphism and polygyny are really changing which kinds of peptides are being bound by having different preferences for MHC anchor residues. And then that typically is forcing hydrophobic side chains to be presented to the surface of this, on the surface of this MHC, and that the act of covering that up with a TCR contributes to antigen recognition. Okay, so I'm happy, I realize this is a lot of material that typically you don't see this kind of biology in other systems. So I'm happy to stay on the line and answer questions you have. But first I want to present the bonus question. Okay, and this is sort of going back to T cell development, and it's often misquoted or misunderstood by immunologists, even today, that they often will incorrectly say that the immunoproteasome has a different cleavage pattern than the non-inflammatory proteasome. And in fact, this is not true. It's actually increasing the efficiency of the process, where it's taking in stuff that doesn't have to be ubiquitinated, and it increases the efficiency of cutting the peptides, but it's not changing how it cuts fundamentally. And the question is, in terms of central tolerance during negative selection in the thymus, what might happen if the specificity of proteasomal cleavage changed during inflammation? So if suddenly you start cutting the peptides differently, what problems with central tolerance might you have? Okay, so that is a bonus question. It should be available on Canvas, I believe, in about 10 minutes. Okay, So I'm happy to take questions on MHC1 processing and presentation.
Dr. Blattman, what was the pathway that you were saying about cross-presentation?
So this is where you have to take stuff from the extracellular environment and put it into cytoplasm so it can be degraded by the proteasome. And so you're taking exogenous antigen in through the endosomal pathway, you're feeding it into the cytoplasm by this Sec61 protein, which is sort of the reverse TAP. TAP takes stuff from the cytoplasm and puts it into the ER. Sec61 will take stuff from the ER or the endosomes and put it into the cytoplasm. So it adds one extra step of feeding stuff into the cytoplasm, and then it's just a normal pathway of proteasomal degradation and refeeding it back into the ER to bind to class 1.
So it's kind of like a two-step where it goes into the ER twice.
Right. It's first brought in this way from the outside the cell, put into the cytoplasm, degraded, and then put back into the ER.
Is that similar to one of our, I can't remember, one of the pathways that we had learned in the first part of our lectures? I can't remember where we were using like the complex proteins. That's not a complex protein.
The complement proteins?
Complements, thank you.
No, complement proteins are outside the cell. So they're not aiding.
Then it's not brought in, okay.
So I think what you're thinking of is complement receptor 1 versus complement receptor 2. Those are not really feeding into this pathway. They're more important for transporting antigen to the lymph node to generate a response.
Okay.
Dr. Blattman?
Mm-hmm.
Did you say that this pathway only occurs in professional APC cells?
Yeah. So it's cross-presentation is important for activation of T cells. It's not really, you don't want cells cross-presenting and getting killed if they're not infected. So it's really only by professional APCs.
Do those also do the classical cytosolic pathogen pathway as well?
If they become infected, yes. Remember, cross-presentation is just adding an extra step. You're just feeding stuff into the cytoplasm. So if it's infected and it has proteins in the cytoplasm already, that pathway is still going on.
So these happen side by side?
Correct.
Okay.
When I first took immunology, the idea of cross-presentation was not really appreciated, even though it had been discovered. And so the textbooks at the time all said that, well, in order for activation of a CD8 response, you had to have a dendritic cell that was infected, which didn't make a lot of sense because, well, a virus would just not infect dendritic cells and then you'd never make a T cell response to it.
Can you go over the difference between polygyny and polymorphism for time?
Sure. So polygyny is just you express multiple copies of the allele. So you have, let's start off, if you only had one MHC, okay? So at most you would have 2 different chances of presenting peptides from a pathogen. But now you have 3 copies, or you have 3 alleles, or I should say you have 3 MHC1 versions, and they're all codominantly expressed. So you actually have 6 different potential ways of presenting an antigen to a T cell. So that means you have a much higher chance of actually being able to present something from whatever's infecting you. Polymorphism means that in a population, there's many different versions of each of those alleles. So polygyny refers to you're expressing many different or multiple MHC proteins. Polymorphism means that there's lots of different ones, and so in a population, you increase the chances that somebody in the population is actually going to make a response and survive that infection. Another way to say that is polygyny means you have HLA-A, B, and C. Polymorphisms is it's A02, A03. Sorry, my handwriting's not very good with the pointer here. But it means that for HLA-A, there's many different versions of HLA-A. Polygyny means you have 2 copies of HLA. You have HLA-A on one you go back here on one chromosome and you have another copy of HLA-A on the other chromosome. And so you get, out of all the different polymorphisms in the population, you get to pick 2. You get to pick 2 HLA-B, and you get to pick 2 HLA-C. So this gives you 6 total. But in a population, there are hundreds to thousands of different HLA-A versions. And so you only get to pick 2, but the population, there's many polymorphisms so that individuals have very different selection. They may pick 2 entirely different ones than you.
So you can have like an A1 on one chromosome and like an A3 on a different chromosome.
Correct.
Okay.
But in the population, you can have A23, A47, A51. You can have lots of different versions. Other questions?
I have another one if no one's gonna ask.
Yeah.
Can you go over the pathways of the chaperone proteins again?
Sure. So you're talking here?
Yes.
Okay, so step 1 is just, you got to make a new MHC. And so, excuse me, calnexin is the protein, the chaperone protein that just allows you to feed this through into the ER and fold it properly. Okay, the ERP57 and calreticulin act together to hold that newly made MHC1 open, where tapazin brings that MHC1 next to the tap protein. Okay, so you have to fold it, then you have to hold it open and put it next to tap. Once you have that, the peptides that are being fed through the tap will bind to the MHC, and that causes a conformational change in the MHC protein, and you can then, it no longer binds those proteins, the chaperone proteins or tapazin, and it's released to go to the cell surface.
Is there anything that transports those to the cell surface?
There are, but I don't require you to know those for this course. It is the default membrane-bound protein pathway. So it's actually going along the microtubules here. It's being actively transported to the cell surface, but I think going into that level of detail is not really necessary for here.
Okay.
Are we gonna need to remember the different like the alpha beta domains that are on MHC class 1 versus class 2?
Where they occur, you mean?
It's like it says 2 types of MHC proteins, that slide. You have like MHC class 1 has alpha 1, alpha 2, alpha 3, and then beta 2 microglobulin. And then MHC class 2 has the beta 1, beta 2, and alpha 1, alpha 2.
Correct.
Are there different functions to that we need to know?
Well, no. The alpha 1 and alpha 2 are primarily for antigen binding, right? This has the alpha helices on the outside, and that corresponds to the alpha 1 and beta 1 for MHC2. So here's alpha 1. Somebody needs to mute themselves. It's pretty loud. The alpha 3 and beta 2 are just structural. The alpha 2 and beta 2 are just structural. So that's why they're colored the same way. They have essentially the same role. So the question is, why does the immunoproteasome not require ATP? So we go back to this step, right? Under the normal proteasome, the 19S regulator, the cap of this, only recognizes proteins that are ubiquitinated. In order to get ubiquitination, you need ATP. Okay, so that's under normal conditions, you'd need ATP to feed stuff to the proteasome. But during infection, ATP becomes limiting, and so if you still require that, proteins are not getting ubiquitinated because there's not enough ATP, so you wouldn't be able to feed those to the proteasome. And so the new cap in the immunoproteasome just recognizes any amino acid. It doesn't have to recognize ubiquitin, and so there's no requirement for ATP because you don't have to ubiquitinate proteins that are going into the immunoproteasome.
And that's just because there isn't enough ATP that's being processed during an inflammation.
Well, it's actually, if that cell is infected, it is, the pathogen is using up all of the ATP to make more pathogen. And so infected cells have a deficiency in ATP. They're energetically, they don't have enough to do this. So you would have a deficiency in ubiquitination.
Is that the same if somebody has like mitochondrial syndrome?
It can be, yeah. So in mitochondrial syndromes, you typically don't make a lot of NADPH, which then is converted into ATP. And so it essentially can have the same thing. But if you have mitochondrial syndrome, you're typically in inflammatory states much of the time. Okay, any other questions? Okay, if there's no other questions, I will see you all on Thursday.